I am very pleased to introduce our next speaker, Elena Dragicevic. Elena is a Senior Sales and Alliance Manager at Nanion Technologies. Before joining Nanion, Elena completed a PhD in neuroscience in Munich before moving to postdoctoral studies in neurophysiology and actually running an electrophysiology lab at the University of Ulm. Welcome, Elena.
Okay, perfect. So I'll just go down with the questions. So thank you very much for the invitation, even though if it's virtual. I will start with the first slide that is some kind of basically an overview slide. It goes a little bit in the future, it goes in the past. In the middle here, you can see let me see if I take a little pointer. So what you can see here in the middle is basically the current S7B guideline. And for this guideline, multiple different considerations have been taken into account. As you can see here, the integrated risk assessment consideration between non-clinical and clinical studies, as well as the best practices for both electrophysiological experiments, as well as the different myocyte assays, which you can see here on the left side. And the reason why they were all made is to basically make the best possible in vitro practices, which should have standardized and harmonized approaches in order to reduce different data variability, also to be comparable across different labs, and also to enhance this translation to clinical findings. And the reason why I'm showing this is my next slide, and those are the ongoing studies as part of the HESI cardiac stem cell working group. And the new functional endpoints considered are actually different electrophysiological points, such as data that would come from ion channel research, for example, axial potential duration, or coming in from the contractility. And we have, together with FCDI, worked on multiple different projects where we have basically integrated ion channel physiology research in iPSC-derived cardiomyocytes in virtual or also mechanically induced pro-maturation environment. And for this, we have used two systems, well, three actually, but two would be the focus of this talk. And the first one is represented here. So this is the patch liner with an add-on dynamite. And this system basically allows to record in an automated dynamic clamp mode. It records in 8 cells simultaneously. And what it does, it basically enables a simultaneous recording of stable negative resting membrane potentials, and also improves the stability and shape of the action potentials recorded from iPSC-derived cardiomyocytes. The second one, the one below, the FLEXcyte 96, is actually an add-on to an impedance system and MEA-like system, CardiacCyte 96. And what that one does, it basically allows the cells to grow in a more in vivo-like environment. So basically the flexible membrane that it's characteristic for this system is mimicking these mechanical conditions of the heart. So we could enhance the evaluation of different drug candidates, for example, for cardiac safety or toxicology or efficacy studies. So I will start with the automated patch clamp, and for this, as I mentioned, we have used the patch liner system, and this system basically works in a way that the robot that it's integrated in the system is pipetting the cells and solutions on top of your cells. And if for people who haven't used a patch clamp before, normally the patch clamp works in a way that the pipette is being brought to the cell membrane where the seal is being made, and through different techniques, one can basically take a peek inside the cell and see which ion channel currents are being active or not active in those cells. Here one does that with a robot, and the idea is that the cells are randomly caught inside of this chip. The seal is made, and the external and internal compositions or basically solutions can be exchanged. So if we look at the iPSC-derived cardiomyocytes, there the key factor for the maturity was also the presence of the IK1, and cells that are not that mature do not express a lot of this ion channel. In order to compensate for that, we have added the Dynamite 8, which is the dynamic clamp mode for the patch liner. And as you can see here, the black would basically be the action potential with only holding current. The dark blue would be the action potential as 75% seal compensation. And the light blue is basically the action potential with 75 seal compensation plus the injection of the IK1, and it was in this case 700 picosiemens per picofarad. This is the value that we have experimentally found it was the best. So as you can see here, there's a very nice shoulder action potential shape when recording in this mode. What we have done further is we have also looked at the presence of the ion channel currents in iCell squared. And we were able to record the sodium currents, outward potassium currents, and we also see more ventricle type of cells. So the potassium currents are rather small. And also L-type calcium currents were present as well. When we recorded in different SIPA protocols relevant to the first slides that I mentioned, this is just one example. So this is the sodium late current, but you can see here, it's a very small current that can be activated with ATX2 and blocked with renolazine. So this is exactly what we have done. You see here in dark blue the activation, and with light blue the block. And this can also be shown here in the scatter plot as well as in the bar graphs. So with this, we see that we can generate very nice amplitudes of this current in iCell cardiomyocyte squared, and we could perform the SIPA protocol in a highly successful way. This would then satisfy the SIPA criteria for this assay. What we have done further is now look at the dynamite, and this would be the dynamic clamp recording of the action potential. This is one of the representative traces, and what you can see here is basically the data that we have observed from 8 cells. So those are the 8 out of 8 action potentials which could be elicted. We have had around 75% success rate in this system. And what we've done there as well is look at the different pharmacologies of different ion channels. So in this case, this is the calcium channel. We have used BK and nifedipine. So with BK, we have been stimulating channels, as you can see here. So we could increase the shoulder of the action potential by, or prolonging the action potential by increasing the concentrations of BK. For this, we were able to see the entire full concentration response curve. And we were able to block this effect with nifedipine as well. Additionally, we have also done the experiments for the potassium channel pharmacology. So in this case, was the fetolite was applied. So similar experiment as before. So with the application of the fetolite, we were able to prolong the action potential duration, and by washing it out, return to the basic or control conditions. So now I will also mention a little bit higher throughput. So what I said now was 8 cells that could be recorded in parallel. Another system, SyncroPatch 384, is able to record 384 cells at the same time. And usually these types of assays, and also during the SIPA study where we're doing them in these heterologous cells, like for example, HEX or CHO cells, but we have also tried to record I-cell squares on this system. And as you can see here, we used 128 wells. From 100 wells, we were able to record 99% sodium currents, 56% calcium currents, and 93% potassium currents in these cells. So with that, I will switch to the contractility area of or part of the talk. And what we can see here, and for some reason the movies are not going, so let's see like that. So what you can see here is basically the bottom of the plate. So those are the wells that are full with media and have cells on the membrane. And by contracting, like you can see here, these are the I cells squared at day 4 after seeding. The cells are basically lifting this membrane, and by changing the capacitance field in between the sensing electrodes and the membrane, we can calculate the contractility of the cells. And what we've done here is we have also tested different compounds. So to have a little bit similar analysis as to automated patch clamp, we have tested calcium and potassium blockers, as you can see here on this slide. On the left-hand side are the data from nifedipine. These are the raw data, so how the traces look like. By adding nifedipine in increasing concentrations, we have seen the increase in the beat rate with increased concentration until the 1 micromolar concentration, and the amplitude was going down. And for the dofetilide, we were able to detect arrhythmias, which are marked here in the gray areas. The more important for this type of experiments are actually the inotropic compounds, and for this we tested a lot. We did some here at Nanion, and some we've done with our collaboration partners in vitro. In this case, we show, or I showed 2. For on the left-hand side, you could see omecamtiv mecarbil data, and on the right-hand side, the SBK. Both are inotropic compounds, but they have different mechanisms of action. So if you would look here on the left-hand side for omecamtiv, we could see the increase of both the concentration amplitude as well as duration of the contraction relaxation cycle, so the beat duration. And this would indicate the positive inotropic effect, which is based on the activation of the myosin complex. For this, we did not see any change of the symmetry of the signal, but on the right-hand side, you can notice the change of the symmetry. So this would be the effects of the SBK. In this case, we have the contraction phase, which is unaffected, but we have the prolongation of the relaxation phase, and this would be the result of basically increased calcium concentration, intracellular calcium concentration inside the cell in this case. What we've also taken a look at were different chronic cardiotoxic effects. So the previous ones were acute, so we just applied for maybe 1 hour and then did the analysis at minute 15. In this case, we were looking at the longer applications. So in this case, on the right-hand side, you would see a few of the kinase inhibitor substances, which we have observed over the 5-day period. For them, we could see these cell arrhythmias as well. And here in the gray area, you see the detection of the different kinds of the arrhythmias as well. And this we have also performed in collaboration with in vitro. What you see here is a little bit more detailed description of the experiments that have been done on one of these kinase inhibitors, in this case, Gefitinib. And what we could see over those 5 days is the significant functional cardiotoxic effect during the subchronic exposure, which would be here listed in these graphs, depicted in the normalized amplitude going down and the normalized beat rate going down in high concentrations. What we have also looked at are the effects of, in this case, erlotinib and Vandetanib, which are the kinase inhibitors and anthracyclines such as idarubicin. And these were experiments that also lasted for 5 days. And with erlotinib, which is considered generally cardiac safe, we have performed the experiments that basically prove or show this, this claim to be true. The only time that we have seen different effects were at higher doses, so which would of course exceed the clinically applied dosages. This also shows a negative inotropic effect of these compounds. For Vandetanib, we were able to replicate this, basically the toxic effect of the substance. It shows that the effect was not time dependent, but it was dependent on the dosage that was added to the cells. And for idarubicin, we have seen both dose and time-dependent effect in the cardiotox. What we have done additionally is the stimulation of the cells. So if you look at if we just think about the picture that I shown before and how basically how the plates look like, they have this flexible membrane and they don't have electrodes at the bottom. So electrical stimulation gets a little bit difficult. And for this, we have tested the optical stimulation, and for this we used the optical add-on called SOL, which would stimulate at 47 nanometers. This was done in 30 seconds simulation sweeps. And what you see here on the right-hand side is basically the stimulation rate in plotted against the beat rate, so the one that we want compared to the one that we would like to see. And you see here that they correlate very nicely. On the upper side, you see the example traces from 1.75, and 2.25 hertz. Additionally, we wanted to stimulate the cells in another way, and that would be a way that basically would mimic the in vivo situation. So if the heart would be contracting, it would be changing and stretching its cells. And for that, we have been working on an active pressure regulation prototype. So this would be a modified CardioToxic 96. And for this, we are generating the system that it's able to apply cyclic and constant pressure to the silicon membrane where the cells are placed. For this, we've also worked with iCell cardiomyocyte squared, and we're able to elect an elastic modulus of around 30 kilopascals. For this, we have done 2 types of experiments. We have done applied different levels of constant strain to induce membrane deflection, or basically to reproduce the Frank-Starling mechanism. And the second would be a 1 hertz cyclic pressure stimulation, which was induced by 5% membrane deflection, and this was applied for around 3 days. So in order to test the Frank-Starling curve or mechanism, we have increased the levels of constant stretching, and they were applied to the cell monolayer via the pneumatic pressure I mentioned before. So during our experiments here on the left-hand side, you can see that we have observed that the contraction amplitude was increasing under higher strains in both the serum-free and maintenance media. And below, you can see the raw data. So basically the levels or the data coming from the FLEXcyte for different percent of strain with serum media and the maintenance media, one next to each other. With this, we could basically successfully reproduce the Frank-Starling mechanism inside of the system, where by applying higher steps of pressure, we were able to induce the higher membrane strain and higher contraction amplitude of the cells. The second experiments that have been done are the cyclic pressure stimulation. So with this, one would basically look at the 5% cyclic stretching that have been done after 3 days of plating. In the blue trace, you see the stimulated trace. In the gray trace, you see the controlled. So you could see the change in the form of the trace. And the morphology was different, which you can also see in these scatter plots showing the different parameters. First of all, also the amplitude was increased, as well as the upstroked velocity and the relaxation velocity, including the rising and falling time of the contraction. So with this, I would like to conclude my talk, and I would like to thank the people who are thinking outside this cardiac box. Those are the group who actually did the experiments. So Andras, Marta, Ulrich, and also thank to InnoVitro for their part, to Matthias, Peter, and Bettina. And many thanks for FCDI for the opportunity, and also for the very nice work together to Ravi, Simon, and Kara, who helped us a lot for developing the protocols for this data. And thank you very much for your attention.
Thank you very much, Elena. Wonderful talk. So if we have some time for questions, let's see. So I guess going back to the patch experiments, we were curious about, have you looked at the IKs? Can you measure those with the iSol cardiomyocytes as well?
So the currents that I've shown are the basically is the bulk potassium current. So we have not used any blockers. Okay. But yes, if we could basically isolate the current through application of various blockers, we can do that. We are also running a lot more experiments also with the new iCell squared strain. So we're looking forward to have them soon also to show.
Great. Yeah, us too. And then, so are you doing these recordings at room temp or at physiological temperature?
We can do both. So we can do, so for automated patch clamp, we can, well, for both automated patch clamp and also FLEXcyte experiments, we can do both room temperature and 35 or 37 as physiological temperature. In this case, I think the HERG was done on physiological temperatures and the others were not.
And then, so what do you think, what would you say is the main benefit of doing the dynamic clamp?
Yeah, well, for what I mentioned before concerning this maturation factor, basically the less mature the cells are, the less IK1 they have, and that would basically disturb their resting memory potential. So the recordings will not be as stable, and the shape of the signal would not be as good as it is. So by controlling it in iPSC-derived cardiomyocytes, we are able to record better and more reproducible signals. So
turning to the FLEXcyte a little bit, I guess then. So it's really cool to see how that's working with the the membrane flexing and stuff. So can you broadly comment, I guess, on the how does it different to work on the flexible substrate compared to rigid?
Yeah, we were doing a lot of comparisons because we also the add-on FLEXcyte is an add-on for another system, CardiotoxiC, and that operates on a rigid substrate. So it's a substrate that it's just a simple plastic, just like any other normal petri dish or classical plate, so far to look at the mature effects of to get the mature response for the inotropic receptor, inotropic compounds, one would need to stimulate the cells. And I don't know if you noticed for the data that I've shown, or I didn't say it, the cells were not stimulated. So the cells were just plated, and they shown this mature-like phenotype just by being there on this flexible substrate. And we think that the reason, or we postulate that the reason for that is this mechanical environment that gives them the feeling like they're home. So the movement of the cells, it's much more physiological.
Yeah, I mean, I guess they're actually doing a little bit of work, as it were. Yeah. So in terms of that stretching, then, so do you know, is there differences between, I guess, like radial stretching and anisotropic stretching? Have you looked into that?
Yeah, I'm not sure if we have taken a look at this because the system is currently in R&D. So basically, we're developing the applications. But this is something I can, I hope I could give more information next time.
And so there's a couple questions about the optical simulation. So just how generally is that working? Are you just transfecting with the channel rhodopsin, or how does that?
Yeah, the cells were transfected. The experiments were done in vitro. So what they've done, they have transfected the cells with channel rhodopsin 2, and they use the lid basically to stimulate the cells in different wells with different frequencies. So that was the bar graph that I've shown, where the stimulation correlated to the cell contraction.
So you're commenting about the inotropic response. So just when you're doing I guess even before or after the pacing, optical pacing, are you seeing?
Oh, we haven't tried. So this was the first experiment that we've done. So we had a lot of questions about the stimulation. So what the plan is to basically stretch, or maybe that also comes together with a previous question, to stretch or to stimulate optically, and then to see the effects of inotropic compounds additionally, to see if there's also any change.
Right. Okay,
so far it was just the light.
I see. And so similar to that, then, with looking at force frequency relationship, have you evaluated that with or without the stimulation?
So we have done it just so without the stimulation. So it was the isoprenolone response. This was the classical positive response. So I didn't put it in because we showed it so many times. But yes, we were able to do that without the stimulation, additional stimulation. So just the classical, just a normal contraction of the cells.
Okay. Looks like we have probably time for one more question here. So just are you looking at doing different stiffness of the membrane, or is it are you available just in a single stiffness, or that's an interesting thing to look at, I think.
Yeah, I think this was something like we were also thinking about, and I think the guys from Innovitro already tried different ones. I think for us the most easiest thing would be also for the users of the systems at the end is to be able to modify it through the pressure. Yeah, I think they tried this. It's just not something that I have more information about.
Well, great. Thanks so much, Elena. We really appreciate the talk.
Very welcome. Thank
Thanks
you.
for entertaining questions. Yeah.